Okay, so molecular systematics is the key to classifying all these prokaryotes because prokaryotes are so diverse in their structure, function, metabolic, ecological functions. It's really hard to narrow down exactly the categories of them. But they have so far split them into two domains: domain bacteria, which is supposed to be the oldest, domain archaea, which split independently as well, very early on here, and then you have domain eukarya, which is over here. Now, the fossil record is too incomplete due to this early divergence right here in Earth's history for us to really know for sure how to narrow down the phylogenetic tree. But prokaryotes are too simple and microscopic structurally to use for comparative anatomy. So we are forced to use molecular materials like DNA. And particularly what they like to use is the smaller subunit rRNA, ribosomal RNA, because all organisms have ribosomes. So what do you do when you need to find a common ancestor? You find what every single organism on Earth has in common, and that would be ribosomes, because every organism has to have proteins. So they use phenotypic characteristics before. They were using phenotypic characteristics like, is it gram positive, is it gram negative, what shape, what's the motility like, is what kind of nutrition does it, how does it get its nutrition? And these are all useful in identifying pathogenic prokaryotes, but they don't really help very much when you're trying to classify all of the prokaryotic phylogeny. Why? Because lots of these characteristics are scattered across all the groups. So you'll find these characteristics amongst all of these groups, so it's really hard to use those as grouping mechanisms. So they've already narrowed down that archaea and bacteria diverged early in Earth's history. There's a table 27.2, which is good at breaking down and comparing the different characteristics across the domains. So it looks something like this. It's good to know per se, but I'm not going to I'm not going to test you like directly over it, but you should know some of the characteristics of it. So going back to our phylogenetic tree here. You see that archaea has unique characteristics because of its separation, because of its separate evolutionary path that it's taken. You have these extremophiles, which are going to be extremophile means lovers of extreme environment in which other organisms cannot live. All right, so archaea is split up into extremophiles. You have the methanogens, which use carbon to oxidize hydrogen to produce methane, which is CH4, which is a waste. You find this a lot in the intestinal tracts of cows and many types of farm animals. They're obligate anaerobes. They live in swamps, marshes, and the guts of animals. They're decomposers, mainly, of sewage. Okay, so these are the Euryarchaea, all right, which is what they're called. The second type of Euryarchaea, which is here, and it's also an extremophile, are the extreme halophiles, which means that they like to live in very high saline environments. They form a red, purplish like scum due to the bacteria rhodopsin, which is a pigment that kind of gives off like a purpley red color. And that's why the Dead Sea looks like red, like the reddish on the bottom. And the Great Salt Lake and a lot of the high salinity areas of the ocean. So those two, the methanogens and the extreme halophiles, are part of Euryarchaea. Criniarchaea are going to be the extreme thermophiles. All right, these would be the high temperature. They live in very, very hot environments like sulfur springs, deep sea vents, volcanic areas, and these are proposed to be the ones that evolved very early because, as you can see here, they evolved very early because that's what early Earth was like. It was very hot, it was extreme temperatures, and then it would go to extreme cold. So these had to be able to live in these very, very harsh environments. Now, the bacteria here include most prokaryotes, or most prokaryotes are sectioned off into this area right here. They're extremely diverse in their nutritional modes and metabolism, so they've broken them down into sort of 5 major groups. All right, the first one being proteobacteria, which actually includes alpha, beta, gamma, delta, and epsilon types of bacteria. They have a really good table on page 27.3 here. And it goes through all of the bacteria, what all their characteristics are like. I'm not going to make you know all this. I'm going to tell you what you need to know for it. All right, alpha. Alpha, beta, gamma, delta, epsilon, gram-negative, all of them. Alphas are usually parasites or mutual symbionts, like Rhizobium, which is going to be the bacteria that live in the roots of many plants. Beta are going to be more of your nitrogen-fixing bacteria. Gammas are going to be pathogenic enterics, which means that they live in the they live off of tissue, so like E. coli and Vibrio, things like that. Delta are going to be the colony-forming bacteria and the predators of other bacteria. Here you can't see. Here you see that this tiny bacterium is actually going to attack that larger bacterium. And then the epsilons are close to the deltas, but these are the ones that would, I guess the one that stands out the most is Helicobacter pylori, which actually causes stomach ulcers. And you can get Helicobacter, you can find it a lot on the skin of chicken. So if you're eating chicken and you're eating the skin and it's not cooked very well, then you can also get sick off of that. Going back to our little table here, you see chlamydias are next. All right, if you guys know anything about chlamydia, you already know where I'm going with this then. All right, chlamydia are parasites of animal cells. They're gram-negative, and it causes the STD chlamydia. Okay, so, it's sexually transmitted, and it's the most sexually transmitted disease in the United States because a lot of women will not show any symptoms of it, but it is also gram-negative. Next one are going to be the spirochetes here. Okay, these are helical with corkscrew movement. They're free-living. Some of them are pathogens, and these include diseases like syphilis and Lyme disease. All right, by their name, spirochete, you should be able to at least know that they're helical and they move in a corkscrew fashion. The next one, which is a big group, gram-positive bacteria. They are as diverse as the proteobacteria are. Okay, you can see here that they give you a ton of pictures for them. They're some of them are gram-negative, actually. Okay, they're closely related to the gram-negatives a little bit. This includes tuberculosis, leprosy, but most of them, those are the bad ones. Most of them are actually just free-living decomposers. This is where you would these are the ones where we can synthesize antibiotics. This is where we get anthrax and botulism from. And these also include something called a mycoplasma, which lacks cell walls completely. And the last one we're going to talk about is cyanobacteria. We've already heard a lot about these. Okay, these are photosynthetic, autotrophic. They're plant-like. They're oxygenic photosynthesizers, which means they use oxygen. They're solitary, and they can also live as colonial. And they usually live in water, and they are a major, major source of food in a lot of ecosystems. And these are the ancestors of photosynthetic plants and algae.